Welcome to the Entomological Society of Washington annual banquet. It's a great honor for me to be here with all of you, sharing our love for entomology. And before we start, I would like to thank all the members of the executive team, especially Abby Kula, Alisa Seaman, and Lourdes Chamorro. This event would not be possible without them. I also want to thank Cici Escobar and Renan Guerrero for the support. Also, I want everybody to check out this banner. It has some QR codes as a quick and easy way for you to become a member, subscribe for news and updates, and also to make a donation to help the society keep growing like a beehive. Also, as you know, dinner is included, but feel free to tip the people from Busboys and Toys. Now it's my great honor to introduce my friend, the amazing Dr. Ted Schultz. Ted Schultz has worked for over 30 years as a research entomologist and curator of ants in the Department of Entomology of the Smithsonian Institution, National Museum of Natural History. His research is largely focused on fungus-farming ants, a group of around 250 species that began farming fungi for food around 66 million years ago. And he's particularly interested in the coevolution of the fungus-farming ants and the many fungal species they cultivate. Ted has authored over 120 publications and edited and authored 4 books. During the past 34 years, his research has taken him on 32 field expeditions, excavating the nests of fungus-farming ants in 11 South and Central American countries, including 15 trips to diverse locations in Brazil. Ted received a bachelor's degree in biology in 1988 from the University of California, Berkeley, and a PhD in entomology in 1995 from Cornell University. He is an elected fellow of the American Association for the Advancement of Science and an adjunct professor of entomology at the University of Maryland in College Park. Please join me in giving a warm welcome to Ted.
Well, thank you. Can you, is this working? No. Closer. How about that? No? Hello? Yeah. Okay. Let me know if I drift away from the microphone. I'm not good at this. But thank you. Thank you, Toledo, for that wonderful introduction. Thank you. And thank you for inviting me to give this annual banquet talk to the venerable Entomological Society of Washington, which I consider to be a very high honor. Thank you. Okay, let's see if this works. Oh, did that not work? And please tell me if I'm drifting from this microphone. I mean, I could hold it. Then I feel like a lounge singer. You think so? Okay, I'll try. Okay, as most of you know, if you were to travel to Central or South America, is this working? Or even to certain places in the southern United States, one of the most wonderful sights you'll see are leafcutter ants. thousands of ants carrying cut leaf fragments along well-manicured paths back to their nests. Leafcutter ants were no doubt well known to the earliest humans to arrive in the neotropics, but unfortunately, we have no written records to tell us what they learned about them. A notable exception is the Popol Vuh, the creation myth of the Mayan people, which does survive in written form. Leafcutter ants are prominent characters. Let's see. Oh, it's skipping a slide. Oh no, there it is. Yeah. So leafcutter ants are prominent characters in the Popol Vuh. But with the exception of the Popol Vuh, the written record begins with the early Europeans. In the 19th century, the English naturalist Henry Walter Bates published the first detailed written account of the behavior of leaf-cutting ants in his classic 1863 book, The Naturalist on the River Amazons. Bates observed leafcutter ants cutting leaves and carrying the leaf fragments into their nests, and he wondered why. He wrote that the leaves are used to thatch the domes which cover the entrances to their subterranean dwellings, thereby protecting from the deluging rains the young broods in the nests beneath. So Bates thought that the leaves served a waterproofing function, and unfortunately for him, he was incorrect. 11 years later, in his 1874 book, The Naturalist in Nicaragua, another great English naturalist, Thomas Belt, wrote, I believe the real use the ants make of the leaves is as a manure on which grows a minute species of fungus on which they feed, that they are in reality mushroom growers and eaters. Thomas Belt turned out to be right. In that same year, 1874, another great naturalist, the 19th century German Brazilian naturalist Fritz Müller, independently discovered fungus cultivation in ants, communicating his discovery to the very young journal Nature through his correspondent, who happened to be Charles Darwin. As to the leaf-cutting ants, I have always held the same view which is proposed by Mr. Belt, namely that they feed upon the fungus growing on the leaves that they carry into their nests. So that means that last year, 2024, was the 150th anniversary of the discovery of fungus farming agriculture in ants. So I hope everybody celebrated as much as I did. Just to make sure the 150th anniversary was not overlooked, 4 of us wrote a letter to Nature pointing out that Fritz Müller's 1874 letter to Nature was conveyed by Charles Darwin, the founder of evolutionary biology, and that Thomas Belt's 1874 book, The Naturalist in Nicaragua, was reviewed in Nature in 1874 by Alfred Russel Wallace, the co-founder of evolutionary biology, and that we've learned a whole lot about fungus farming and agriculture in the subsequent 150 years. And this evening, I hope to summarize some of what we've learned. Human agriculture originated at least 14 and as many as 23 separate times in human history, usually with multiple crops per origin. This includes the many origins of seed crops across the globe, and it includes the many origins of vegetatively cultivated crops across the globe, crops cultivated by cutting. Less well appreciated, agriculture also originated about 20 known times in non-human animals. This includes agriculture by damselfish, polychaete worms, snails, but most relevant to those here tonight, by insects, fungus-farming termites, fungus-farming beetles, and fungus-farming ants. 3 years ago, 3 of us, 2 biologists and an anthropologist, edited this book in which we drew parallels between insect and human agriculture, and in which we regard agriculture as a mutualism. In biology, mutualism is a relationship between 2 or more species that benefits both or all of the participants. You can contrast that with another kind of symbiosis, parasitism, in which one partner benefits at the expense of the other. We also propose that agriculture is a specialized form of mutualistic symbiosis. You could say that our domesticates, because they get eaten, do not benefit from agriculture and that it's a parasitism, unless you consider facts like this: the total biomass of extant humans and their mammalian and avian livestock is 17 times greater than that of all the wild mammals and all the wild birds in the world combined. There are similar statistics for our plant crops like wheat, rice, soybeans, maize. So in terms of their relative numbers on Earth, and in terms of passing their genes from one generation to the next, which is the definition of fitness, our domesticates benefit tremendously from their associations with humans. I'm going to get my water. Oh, I can keep talking while I move around. Benefits. Okay. Fungus farming ants include the, did I skip a slide? Oh yeah, I did. So this evening I'm going to talk about one of these agriculture groups, the fungus farming ants. And the main reason I'm going to do it is because they're my favorite organisms on the planet. This is an exclusively New World clade, confined to the New World, 247 described species, and many, many more yet to be discovered undescribed species. All fungus farming ants are obligately dependent on the cultivation of fungus gardens for food. Fungus farming ants include the well-known leafcutter ants, the ants observed by Bates, Belt, and Mueller. Nests of leaf-cutting ants in the genus Atta are huge. Can you see my cursor here? No? Well, notice that trail that the ants have made coming away from their mound, and there's probably many more of them radiating off in all directions. And in this slide, in the bottom, notice how deep this excavation is and how many garden chambers you can see in the walls of the excavation. Here's another excavation where you can see all those chambers. This is a schematic diagram of a nest of the leafcutter Atta volans, in which you can see the thousands of garden chambers. So I can't show them to you with, oh yeah, I can. There it is. There's my cursor. Yeah. These are the thousands of garden chambers. These are the much larger waste dumps. And here's a human for scale. These have been called superorganisms, these colonies. They're considered to be the most highly eusocial of all social organisms. And they are the dominant herbivores of the New World tropics. An Atta leafcutter colony is the ecological equivalent of a large herbivorous mammal like a cow. In terms of collective biomass, the total weight of all the ants, which can number 5 million. In terms of lifespan, a colony can live to be 10, 15 years or more. And in terms of the amount of fresh vegetation the colony harvests and consumes every day through its fungal intermediate. These Atta leafcutter nests constitute such a huge and reliable resource that they support entire agro-ecosystems consisting of hundreds of endemic species. Here's a list of all the commensals that Waller and Moser found in a single leafcutter nest that they dug up in Louisiana. And Navarrete Heredia listed 411 beetle species and 25 beetle families, all from leafcutter nests. But as discovered 151 years ago, leafcutters are not directly consuming the leaves. Instead, their garden fungus is pre-digesting the plant matter for them. And here's another one of those trails that the ants have made through the forest. What Bates, Belt, and Mueller didn't know is that most fungus farming ants are not leaf cutters. Most of these little known species are small and inconspicuous. They can have nest entrances like this little turret, and I don't know if you can see, but there's a little worker ant guarding the entrance. And they can have, their fungus garden can be a single chamber in the soil and quite small. Here are some more excavated garden chambers of these so-called lower fungus farming ants. We spend a lot of time in the field excavating nests like this, which I'll talk more about later. Here's a fungus garden that the ants have cultivated on the bottom, the underside of a rotten log, and I've peeled away the bark here to expose the garden. And here are some other gardens on a rotten log, kind of exposed, except that they've been built under a kind of a rain shelter. In both the leaf-cutting ants and the non-leaf-cutting fungus-farming ants, the suite of behaviors required for cultivating fungus gardens is remarkably complex. Ants have to forage for the appropriate organic substrates. In the lower fungus-farming ants, this would be insect frass, seeds, small flower parts, the occasional insect corpse. They have to incorporate that substrate into the growing edges of the garden. They have to go off and find healthy growing mycelium elsewhere in the garden, bring it back and plant it on that newly added substrate. They manure the garden with their own fecal droplets, and those fecal droplets contain high concentrations of proteolytic enzymes that originate not in the ants, but in the fungus. They have to weed the garden. They mechanically remove unwanted bacteria and fungi. They have to remove the spent substrate and get rid of it. They often dump it outside the nest. They have to harvest the fungus for food and feed it to their larvae. And somehow the fungus in a mother's nest has to make it into her daughter's colony. Okay, remember this guy, Fritz Mueller, the co-discoverer of ant agriculture in 1874? He looks like he'd be fun to do field work with. Well, Uncle Fritz had a nephew, the German mycologist Alfred Mueller. In 1890 to 1893, 16 years after Uncle Fritz's discovery, Alfred Mueller traveled from Germany on a postdoc to visit Uncle Fritz in Santa Catarina, Brazil, and to carry out a seminal 2-year study of the fungal cultivars that remains to this day a scientific classic. He described micromorphological characters of fungal cultivars and of other fungi in the garden. Mueller also collected, photographed, and described as a new species these mushrooms, which he found growing out of an abandoned leafcutter nest. And he named this fungus Rosides gongoliforus. It's gone through a few genus assignments since then, but remember that name, gongoliforus. Fruiting bodies occasionally, mushrooms, fruiting bodies occasionally grow in laboratory cultures of ant-cultivated fungi. And based on these very rarely collected fruiting bodies, past attempts to identify the ant-cultivated fungi were generally not very successful, with a few exceptions. As was first figured out in the early 20th century, when a daughter queen leaves her mother's nest, she carries a small bit of her mother's garden in her infrabuccal pocket. This is a pocket found in the mouths of all ants, not just fungus-farming ants. So this huge atta queen started her garden with fungus that she carried from her mother's nest in her mouth. She flew, mated, dropped to the ground, shed her wings, excavated a chamber in the soil. Then she spit out that little fungus wad that she brought with her and took care of it, manuring it with her own fecal droplets, feeding it with non-viable trophic eggs until her first brood of workers emerged and took over the farming duties and also began to forage. About 25 years ago, my colleague Cameron Curry discovered that ant agriculture has a crop disease, a clade of ascomycete fungal species that are found only in fungus gardens and that have co-diversified with the ants and their cultivars since the origin of fungus farming. The disease is called Escovopsis, and it competes with the ants to consume the fungus garden. Cameron also discovered that fungus farming ants culture actinomycete bacteria on their bodies. These bacteria produce antibiotics that the ants use to control the crop disease, Escovopsis. The ant actinomycetes are part of a larger group that includes the genus Streptomyces, from which many human antibiotics have been derived. The actinomycetes are housed in specialized integumental crypts on the ants' bodies that are lined with glands that presumably provide nourishment for the bacteria. We were able to observe these crypts in fungus farming ants preserved in Dominican amber, which is at least 15 million years old. So ant agriculture is minimally a four-part symbiosis with three mutualists, the ants, their fungal cultivars, and the antibiotic-producing actinomycete bacteria, and one parasite, the Escovopsis crop disease. Additional consistently occurring microbial symbionts continue to be discovered in ongoing research. Because researchers have always wanted to know how ant agriculture evolved, they have attempted to reconstruct fungus farming ant phylogenies, evolutionary trees, for over a century. Here are two surprisingly prescient phylogenies by past ant researchers, one from 1912 and one from 1963, based mostly on intuition, but very accurate intuition. Here are some phylogenies based on ant morphology more quantitatively from 1995 and 1998. And here are some early DNA-based phylogenies from 1998 and 2008. Attempts at fungal cultivar phylogenies have been more limited, and they really only began when DNA data became available. Here are two of the first molecular phylogenies of the fungal cultivars from 1995 and 1998. And here's one from 2012. So when we juxtapose the phylogenies of two different groups of organisms in order to study coevolution between them, we call it a tanglegram. In this 2015 tanglegram, the ants are on the left and the fungi are on the right. And the ant tree is based on 4 genes, but the fungal tree is based on 1 gene. The problem with all these early fungal phylogenies is that the few genes that were available could resolve these, they could resolve these closely related groups in the colored boxes, but they could not tell us how those groups were related to each other. Nonetheless, as early as 2005, we could see a pattern in which broad phylogenetic groups of ants, monophyletic or paraphyletic, first tree here, cultivate broad phylogenetic groups of fungal cultivars, middle tree, that are parasitized by broad phylogenetic groups of Escovopsis, the crop disease. We could also see that although ants from one group were never found cultivating fungi from another group, within these groups, the pattern breaks down so that within each of these 5 broad groups, closely related ants can be growing distantly related fungi, and the same fungal species can be cultivated by distantly related ants, but only within the same group. We refer to these groups as agricultural systems, and we would very much like to understand what biological mechanisms enforce the boundaries between them. And just so you know, this pattern of 5 agricultural groups is based on a lot of sampling by a lot of people from a number of labs in the US, South America, Europe, over a 35-year period. This was the approximate number of samples when we tallied them in 2015, and there's been a lot of additional collecting in the decades since then. I wouldn't be surprised if these numbers haven't tripled. And even though we don't know the mechanisms that prevent ants from one agricultural system from cultivating fungi from a different agricultural system, we do have some understanding of why this pattern breaks down within each of the systems. It's because of horizontal transfer. Lab and field observations and experiments have shown that ants can occasionally replace their resident fungal cultivars with cultivars obtained from other ant nests or from feral cultivars growing in the leaf litter unassociated with ants. This could happen if, say, the colony's garden dies, or if a queen fails to bring fungus with her from her mother's nest. Here are photographs of some of these feral cultivars, fungi that can be cultivated by ants, or alternatively, live unassociated with ants digesting leaf litter. So this horizontal transfer violates the pattern we would expect to see if fungi were always transferred vertically from mother to daughter nest without exception. If that were the case, we would expect to see sort of highly congruent matching fungal and ant evolutionary trees, but that's not what we see. I am very pleased to tell you that in the past decade, there's been a revolution in the way we reconstruct phylogenies. For ants, we have phylogenomic markers, ultra-conserved elements, or UCEs, that generate massive quantities of data from loci sampled from across the genome. An earlier version of these markers produced this fungus-farming ant phylogeny in 2017, in which virtually every node is maximally well supported, essentially nailing down fungus-farming ant relationships. Our primary task since then has been to continue to add ant species to this phylogeny using an expanded set of markers. And now, more recently, we also have phylogenomic markers, UCEs, for the basidiomycete fungi. The first and so far only paper to utilize these was published last October, and I'll try to tell you what we found out. These markers produce this phylogeny for the fungal order Agaricales. It's constructed from 475 fungi, including 288 ant-cultivated fungi, all of which arise in just two families, the ant-cultivated fungi, in the Pteriolaceae, the pink here, and in the Agaricaceae, the dark yellow here. And then within the Agaricaceae, they all arise in one tribe, the Leucocoprinae. The Agaricaceae includes the common grocery store mushroom, cultivated and eaten by humans. I'm not sure that's a coincidence. This is the very first well-supported fungal cultivar phylogeny, and it tells us finally how these groups of ant-cultivated fungi are related, and because it's a chronogram, a time-calibrated phylogeny, when these groups arose. Okay, in this tanglegram, this tanglegram includes a simplified version of the fungal phylogeny on the left that focuses just on the ant-cultivated fungi. And on the right is an ant phylogeny that's constructed from 276 ants. And in the middle are lines connecting the ants to the fungi that they cultivate. You don't need to see, and I know you can't see, any of the details here, but hopefully what you can see is this pattern of pronounced fidelity between broad groups of ants and broad groups of fungi, corroborating those agricultural systems I mentioned earlier. But now we know how those agricultural systems are related to each other. This tanglegram indicates that the first fungus-farming ant, the arrow on the right, cultivated two different but closely related groups of fungi, indicated by the red arrows on the left, two distinct, different yellow box groups. We previously knew about these and called them clade 2 and clade 1, but we didn't know that they were cultivated separately and simultaneously at the origin of fungus farming. So the ants that cultivate these groups in the yellow box, paraphyletic group of ants, they're called lower fungus farming ants, and they look, I'll just show you pictures of the faces of a few of them. So here's a couple of faces of lower fungus farming ants. Here are a couple more, and here are a couple more. And lower fungus farming ants typically have kind of smaller colonies with maybe dozens to a couple hundred individual workers. They can have a single garden chamber like this one, or like this one. We know of 4 cases in which a free-living mushroom was collected that when its DNA was sequenced, proved to be conspecific with the same species as or very closely related to a known ant-cultivated fungus. This suggests that although the ants cannot live without the fungi, the lower fungal cultivars are capable, at least temporarily, of living without the ants. So they appear to be facultative symbionts. From generalized lower agriculture, we get a number of evolutionary transitions to different, more specialized forms of agriculture, such as this transition to coral fungus agriculture with the red arrow there. In the ant genus Apterostigma, which I've drawn a red square around, some species cultivate lower fungi in the Leucocaprinae, the yellow fungi, just like all other lower fungus farming ants. Here's one of them, Apterostigma auriculatum, on its lower fungus garden. But around 20 million years ago, the ancestor of this group in the colored box transferred its agricultural behaviors to start to cultivate coral fungi. Coral fungi in the family Pterilaceae are very distantly related to the Agaricaceae that all the other ants cultivate. Humans use agricultural knowledge to cultivate new crops all the time, but this is the only known case of a shift to a completely new cultivar in the ants. And the coral fungus cultivars, although closely related, are actually the products of 2 independent associations with ants. In one of the coral fungus groups indicated by the arrow here, Myrmecopterula velohortorum, the gardens are enclosed in a protective tent-like veil that is woven by the ants from the extremely elongate aerial hyphae, which allows these ants to build their nests in unusual exposed locations, like here on the trunk of a tree and here on the underside of a leaf. But in the other group of coral fungi, Myrmecopterula nudihortorum, there is no veil. This is what those kind of gardens look like. Another evolutionary transition away from lower agriculture occurred in yeast agriculture in the green boxes. So the yeast farming ants and their yeast fungi. Unlike the typical white cottony mycelium that you see here, in yeast agriculture, ants cultivate a very different growth form in which the garden consists of these little yellow nodules. Most fungi typically grow as mycelium, linear branching chains of connected cells, but fungi growing in the yeast-like phase are clumps of unconnected, single, or maybe a few connected cells. Except for the fungus farming ant yeast gardens, yeast phase growth in the entire fungal order Agaricales is unknown. Here's another example of these very interesting yeast gardens. As in the lower fungal cultivars, there have been 5 known collections of free-living fungi that, when DNA sequenced, fell within the yeast fungus clade, suggesting that in yeast agriculture, the fungi are also capable of living apart from the ants, at least temporarily, and that when they do this, they grow in the leaf litter as mycelium rather than as yeasts. They also revert to mycelial growth when cultured on agar in the lab. And in ant nests, they grow as mycelium on the bodies of the ant larvae. So you could argue that the yeast fungi are not adapted at all for life with ants, and that instead they are passively manipulated by the ants, which do something to induce yeast phase growth. And that's possible. But if that's the case, why is it that all the yeast farming ants, which form a compact clade of about 20 species, seem only able to manipulate a correspondingly small compact clade of the yeast fungi. I personally think this phylogenetic pattern is more consistent with the hypothesis that the yeast fungi and the yeast farming ants have co-evolved, and that yeast phase growth is a genetic adaptation for life with ants. In human cultivars, we would call that domestication. Well, a clear domestication event in ant agricultural evolution occurred in the transition from lower to higher agriculture. So the transition from the yellow box to the blue box in the fungi, and the same in the ants. Higher agriculture is practiced by the highly eusocial, ecologically dominant leaf-cutting ants. But it's also practiced by 5 other genera of non-leaf-cutting higher fungus-farming ants. If domestication is genetic change in a cultivar that increases its fitness in the agricultural symbiosis, there are a number of clearly derived traits shared by all higher fungal cultivars that pretty clearly qualify as domestication traits. All higher attine fungi consistently express gongolidia, swollen hyphal tips that are produced by the fungus and preferentially harvested and eaten by the ants. You can just make out clumps of gongolidia here on this higher agriculture garden. In addition to gongolidia, higher fungi are also polyploid, unlike lower fungi, which are diploid. And they have increased expression of plant digesting and detoxifying enzymes, many of which are concentrated in the gongolidia. And perhaps most importantly, although higher fungi are able to produce mushrooms and may or may not be able to reproduce sexually, nobody knows. As far as we know, they are unable to live without the ants. So in higher agriculture, both ants and fungi are obligate symbionts. They cannot live without each other. In the past 35 years, my highest research priority has been tracking down rare fungus-farming ant species that have a high likelihood of providing critical new data about the evolution of ant agriculture. Because even as vast tracts of habitat are being destroyed forever, 43% of fungus-farming ant species are only known from their original collections, and that means their fungal cultivars remain unknown, as do almost all other aspects of their biology. My first example of the value of fieldwork is the fungus-farming ant genus Mycetogroicus, which was only described in 2001. In 2008, we figured out that Mycetogroicus was the sister group to the higher fungus-farming ants, but we didn't know what fungi they cultivated. So we didn't know whether they cultivated lower yellow box fungi or higher blue box fungi. So a group of us, to solve this problem, went to Uberlândia in Minas Gerais, Brazil, where we knew one of these species, Mycetogroicus ceradensis, occurs. We located nests and started to dig, and we were shocked to find that the garden chambers of this species are 3.5 meters deep. That's really deep for a non-leaf cutting fungus-farming ant. I think it's the record. So then in 2012, when I was asked to collect more specimens of this genus for a genomic study, I didn't want to go back there. I decided that we would go after a different species in the genus, one that had been collected on the banks of the Araguaia River, and which I therefore thought would have much shallower nests due to the water table. Well, as frequently happens, I was wrong. And we discovered that nests of this species were also more than 3 meters deep, and that the garden chambers are submerged during the rainy season. And they have these very odd garden chambers. You probably can't see it here, but they have all kinds of little sort of blind tunnels going on of them. And of course, somebody needs to figure this out. But this field work allowed us to identify the fungi grown by both species and to determine that Mycetogroicus is a lower fungus farmer. It's in the yellow box. This in turn allowed us to identify on the ant phylogeny, and now more recently on the fungal phylogeny, the precise branches on which higher agriculture arose. This information will guide future genomic work to identify the genetic change that took place in ants and fungi during the transition to higher agriculture. Paramycetophylax brucki is a morphologically hard-to-place fungus-farming ant species that was put into its own monotypic genus by frustrated taxonomists. That means brucki is the only species in Paramycetophylax. It occurs in marginal habitats in the Argentinian dry Chaco, where I had previously looked for it without success. During the past 5 years, I've had the good fortune to team up with Argentinian myrmecologist Priscilla Hanisch, and she ultimately was able to locate nests of this enigmatic species, which she found consistently occurred under trees known locally as Algarobo dulce. Priscilla became the first person to excavate nests of Paramycetophylax brucki. She collected ants and fungi, recorded biological information about this previously unstudied fungus-farming ant species. When we added Paramycetophylax brucki to our phylogenomic ant phylogeny, we got an unexpected surprise. It's the sister species to the yeast-farming ants, having diverged from them around 36 million years ago. This was published in 2022. And then when we subsequently added the cultivar of Paramycetophylax brucki to our phylogenomic fungal cultivar phylogeny, we got an even bigger surprise. The fungus cultivated by Paramycetophylax brucki is the sister of the yeast fungi. So Paramycetophylax brucki is the sister to the yeast-farming ants, and the cultivar of Paramycetophylax brucki is the sister to the yeast fungi. That's textbook coevolution. What this means is, if we want to explore the genetic changes responsible for the origin of yeast agriculture, our search has been narrowed down to 2 branches, one in the ant tree and one in the fungal tree. Apterostigma megacephala, first described in 1999, based at that time on 4 specimens. It's a morphologically very strange species that was clearly important for understanding early fungus-farming ant evolution. We searched for it without success. Then in 2008, colleagues told us of the collection of a single specimen from a leaf litter sample taken in Carajás in Para, Brazil. So this was the known circum-Amazonian distribution of Apterostigma megacephala at that time. My graduate student at the time, Jeffrey Sosa Calvo, went to Carajás and located a population of Apterostigma megacephala. Here's a photograph Jeffrey took of Apterostigma megacephala on its garden. Here's the position of Apterostigma megacephala, that big red arrow there, on the ant phylogeny. It's in the yellow box, and it should be growing a lower yellow box fungus. But when we obtained DNA sequences for its fungal cultivar, we got one of the biggest surprises of our careers. Apterostigma megacephala is the only lower fungus-farming ant known to cultivate a higher fungal cultivar. And it doesn't just grow any of these higher fungal cultivar, it grows the leafcutter cultivar in the orange box, Leucoagaricus gongoliformis, the fungus described by Alfred Mueller in 1893. To see if this was a consistent association or maybe just a fluke in Carajás, because we had many nests from Carajás, we conducted field work in Mato Grosso in 2017 and collected Apterostigma megacephala in 3 locations there. In every case, it's growing the higher fungal cultivar, Leucoagaricus gongoliformis. I believe that if we understood why Apterostigma megacephala is the only lower fungus-farming ant known to cultivate a higher fungal cultivar, we might get some insight into the biological mechanisms that otherwise constrain the 5 agricultural systems. There must be some kind of physiological, nutritional, or sensory barriers that enforce the boundaries between these systems. So I hope you agree that these examples demonstrate the importance of seeking out rare and poorly understood fungus farming ant species and understanding their biology, especially their fungal associations. Now that we can put dates on the phylogenies of fungus farming ants and their fungal cultivars, we can begin to explore questions about correlations between major evolutionary transitions in ant agriculture and major events in Earth history. For example, what does this ant and fungal dated tanglegram tell us about the origin of ant agriculture? Our ant and fungal chronograms indicate that both lower fungus farming ants and the lower fungal cultivars in the yellow boxes originated 66 million years ago, indicated by these vertical red lines on both phylogenies, and coincided with the end of Cretaceous mass extinction event. That's when a meteor collided with the Earth and caused firestorms for days to weeks and shut down photosynthesis for several or more months, or maybe even a few years, during which global mass extinctions occurred. The fossil record indicates that following the meteor impact, detritivorous fungi proliferated. As pointed out by the ecologist Dan Jansen and others, the types of animals most likely to survive such conditions were small semi-subterranean detritivores or their predators, including among the detritivores, invertebrates specialized on locating and consuming decaying organic matter or scattered dormant seeds and insects. Under such conditions, fungus farming ants and leucocoprinaceous fungi would have constituted a formidable mutualism in which the ants located organic materials and the fungi digested them, sharing the nutrition with the ants. This allowed both partners to persist through a period of mass extinctions. Interestingly, the sister group of the fungus farming ants, which by definition arose simultaneously with them, is the subtribe Dacetina. This is a group of specialized predators. Some of the earliest diverging Dacetine lineages look like this, with these remarkable trap jaws for capturing prey. A study conducted in 1993 that concluded that it's very likely that the early Dacetina preyed on Collembola, which are detritivores. So this explains how they might have survived the end of Cretaceous extinction event along with the fungus farming ants. The transition from lower to higher agriculture, in which higher fungus farming ants in the blue cultivate higher fungi in the blue, clearly involved genetic changes in the fungus, making them obligate symbionts. This transition occurred around 30 million years ago on both the ant and fungal phylogenies, which agree amazingly well about this date, 30 million years. Using the first UCE ant phylogeny in 2017, we reconstructed the biogeography of higher fungus farming ants, and we found out, not particularly surprisingly, that they originated in South America. We also reconstructed ancestral habitats on the phylogeny, and we found something much more surprising, that the ancestral higher fungus farming ant lived in a seasonally dry habitat rather than in a wet forest, as generations of fungus farming ant scientists had always assumed. Significantly, the origin of higher ant agriculture, both in the fungi and in the ants, occurred shortly after something called the terminal Eocene event. The terminal Eocene event occurred around 35 million years ago, and it's indicated by these two vertical lines on the fungal and ant phylogenies. And notice how it very closely preceded the origins of higher agriculture. The terminal Eocene event set off a period of global cooling that resulted in the expansion of drier habitats and grasslands throughout the world. In South America, it resulted in the fragmentation of what was previously continuous wet forest. humans have exerted artificial selection on their domesticates by, among other things, isolating them from their free-living gene pools. And one way they've done this is by moving their domesticates away from their places of origin. This repeated pattern in human agriculture led us to speculate that the domestication of higher fungi likely occurred when the ants transported their fungal cultivars into these new dry habitats in which the cultivar was no longer in sympatric genetic contact with an extended population of west forest dwelling free-living fungi. As it became increasingly dependent on its ant farmers, it was subjected to the ant equivalent of artificial selection, causing it to evolve into the ancestor of the higher fungal cultivars. I'm often asked about the benefits of this research, by which the questioner usually means, how does it benefit human economics? Well, one benefit is that at this point in time, many dozens of antibiotics have been identified by studying the fungal cultivars, by studying the crop disease, Ascovopsis, and by studying the actinomycete bacteria. And these antibiotics include antibacterials, antifungals, even antimalarials. A number of promising anti-cancer compounds are currently under study as well. Fungus-farming ants have practiced agriculture and controlled crop diseases with antibiotics for 66 million years, whereas humans have practiced agriculture for less than 12,000 years and utilized antibiotics for less than a century. Likewise, leaf-cutting ants have essentially husbanded local trees and plants for millions of years, stripping them of their leaves, but not to the extent that they kill them. So I think it's likely that studying ant agriculture and understanding the evolutionary and ecological dynamics could have important and practical applications for the longer-term practice of human agriculture and the longer-term practice of human medicine, because we are currently practicing short-term strategies. Another benefit is simply the wonder and awe that we experience when we become familiar with these remarkable organisms. So I invite you to stay tuned for further wonder and awe. I know I will be. And I thank you very much for your interest. Oh, so the lower ones, like everything, this is a very poorly investigated topic, but there have been some studies. Insect frass is a big thing. There's a lot of insects in the canopy dropping frass, and the ants are down there collecting them and growing their fungus on them. Seeds. Small flower parts raining down again from the canopy. Occasional, it's hard for me to believe, but an occasional arthropod corpse, and various other things depending on the species. And leafcutter ants, when the colonies are very young, they're not even producing the cast that can cut leaves very well. So they're kind of go through a phase where they're also collecting stuff like that. Yeah, I missed the last part. What? Oh, I don't think there's any relationship, but when you look at things like actinomycetes, bacteria, ascomycetes in general, but including Ascovopsis, and even the fungal cultivar, although maybe less so, they produce a battery of antifungal, antibacterial compounds, and compounds that serve purposes we don't even know what they are. And basically, there's a whole sort of biochemical prospecting branch of science where they're very some of the researchers are very focused on fungus farming and you know, these symbionts. And they just challenge them. They grow them up and challenge them with various things and see what their action against those things is. And it turns out, in some cases, they have activity against malaria. So I'm not sure there's any real connection. You know, it's not explained why.
Okay, can you say back this question on the microphone?
Oh, okay. Sorry. Yeah, I'll try to do it.
Well, they're active from like the fungus growing within these chambers? Are there any patterns of porosity or anything of that nature conducive to that microhabitat for fungus farming?
Yeah, so the question is there, do we see any patterns of different kinds of soils, correlations between different kinds of soils and fungus farming and use of those soils in general, or maybe species specific ones? There's been some study of this. The short answer is yes. I mean, we know that some fungus, because when we dig them up, some fungus farming ants are only found in sandy soils. And some fungus farming ants really like clay, dense clay soils. But there have been some studies of gas exchange, carbon dioxide and oxygen and stuff in fairly large leafcutter nests and measuring that and kind of correlating it with soil porosity and stuff. But I'm not sure. That's about all I can tell you. There's not any large scale, like explanatory patterns at this point. I mean, it's just another thing. Somebody needs to be studying this stuff. Oh, yeah.
You're in the field, you do these excavations. Do you always collect a colony? Or do you like, just a little bit on when you're in the field and you do your excavation like that? How long does it take? Do you collect the entire colony?
Okay, so the question is, when we do field work, do we generally collect entire colonies? How long does it take to excavate nests and stuff? Over the years, well, from early on, I tried to collect whole colonies. And depending on the species, that can be very quick. Some species, the chamber's like, you know, half a meter or less down, and it's a single chamber. We can dig those up in, you know, 20 minutes or something. We definitely try to get all the workers, if you never can get them all, but and we definitely try to get the queen. We try to get as much of the fungus garden as we can. And then, you know, there's other colonies like those ones I showed that are like 3 meters deep. That's going to take days to excavate. And then there's a bunch in between. There's some with multiple chambers going down. We never excavate mature leafcutter nests. That would like be insane. But baby leafcutter nests are very easy to excavate when they're a year old or less around. We assume they're about a year old. They're single chamber, very shallow, easy to get everybody, and the queen doesn't go anywhere. Some queens run away, you know. So yeah, okay.
The lower and like you mentioned, the nets are very deep and they don't collect leaves. So how do you even find them since the ants are not bringing leaves and it's like a tiny hole in the ground? And I have another question.
Okay, so you're saying in the lower ants have very deep nests and they don't have what?
They don't have ants that are being deep, so it's not like you can follow the ants to the nest. So how do you find the nest?
Oh, that's a really good question, and it's something that affects our lives tremendously that I didn't talk about. Yeah, I mean, when we get to a spot, like I showed you that Myceta groicus nest that was like 3.4 meters deep or something. Well, it took us days to find those nests. Like we go out in the field, we sprinkle cream of rice cream of rice, it's white, it's easy to see an ant when it picks it up and it's wandering. Nonetheless, you spend, I don't know, an hour or more watching this crazy ant, hopefully returning to its nest, going in and out of the leaf litter, and you're following that piece of cream of rice it's carrying. But eventually, it'll disappear down a hole. And often, there's no mound around it or anything. It's just a hole, a tiny hole. You would never have seen it. You flag it, right? Now you know you've got a colony. And when you've got like that team we had a great team of people. When we had 3 colonies, I think it was, then we paired up and started digging. And then in that case, it turned out to be very deep, but in other cases, maybe not so deep. But anyway, yes, it's a big part of it is finding the entrance. I mean, what actually happens, I should say, is you put out cream of rice, you see what you can tell it's a fungus farming ant going off, and pretty much the first one you take it and you look at it and you make sure it's the ant you want. And then you wait for its sister to come by so you can follow her, So it's like, yeah, it's involved.
Robin asked, I'm not very familiar with fungal farmers, but it seems like many have hidden sculpting their heavy body. It's a commonality, any thoughts on why, e.g., are they pockets for carrying into bacteria in their bodies that themselves can be attacked by pathogenic fungi or something like that?
I guess I should repeat that. Like, no, I mean, it's a question about the sculpture of fungus farming ants and what kind of function that might serve. And is it directly related to the fungi or the actinomycete bacteria or something? One of the things that attracted me to fungus farming ants is they're just beautiful. They have very beautiful sculpturing. Some of them are very spiny. Some of them are very non-spiny, smooth kind of. Some of them are in between. I don't know. I showed you a few pictures of faces where they have a very extended antennal scrobe that extends very far up beyond the profile of the head. So for a lot of that sculpturing, the answer is, as usual, yeah, we don't know. We don't know what it serves. But some of the time, we've spent time figuring it out, kind of. So those long scrobes, for instance, a lot of those ants are hit by predators, and believe it or not, things like poison dart frogs and stuff. They sequester, they eat the ants and they sequester chemicals from the ants and make their poison. And you wouldn't think like fungus farming ants would have any special kind of poisons, but whatever, they must have something because some of these frogs specialize on them. Anyway, those ants often have these extended scrobes that they're able to curl up and protect all their appendages. They can curl up their antennae and protect them in those scrobes. They've got these big flanges on their hide femurs. So when they tuck their legs up, if something's trying to get at their legs from the back, that they're protected. So a lot of that kind of sculpture, we sort of understand. Oh, and then the really spiny ones, that's kind of obvious, lizards and birds and things. It's much harder for them to take an ant with a bunch of spines. But otherwise, yeah, yet another thing that somebody needs to figure out and tell me about it.
Have there been any efforts so far to correlate the origin of fungal agriculture in the Athena New World with its origin in the Old World and the macroturf tiny?
Yeah, there's been no. I've mentioned a few times in papers like, and coincidentally, there's the Old World is occupied by fungus farming termites, and I've asked termite people repeatedly over the years, whether those origins could be simultaneous, because if they were, that would be a kind of seemingly pretty significant, right? And I've never gotten in the past the dates that people were putting on the origin of fungus farming termites were quite a bit younger, and they didn't seem to correlate at all. And now I'm not sure what the current thinking is. So, but there's nobody really, it's not like a topic or anything that anybody's, oh, I'm sorry, I didn't repeat the question. The question was about the dual origins of fungus farming termites in the old world and fungus farming ants in the new world, and if there's any relationship for why they divided up the world like that. And that was my answer. Yeah.
One of the advantages of philosophy is you can look for underlying genetic architectures of the trait. Do you think if you have enough sequence data on the ants, you can find something like a gene for fungus farming?
Find what?
A gene or a genetic basis.
Oh. Okay, so the question is, one of the advantages of having genetic data is you can start looking for the genetic basis of various traits. And do we have enough data to find, say, a gene for fungus farming or something like that, genes associated with fungus farming? And the answer is we're getting very close. So when I talked about the markers we now use, they're not really chosen for their function. They're chosen for their conservative. They all contain very conserved core regions that makes it possible for us to amplify them in all different kinds of organisms. But I'm part of a number of genome level studies, including some focused on fungus farming ants and fungus farming cultivars. I'm very excited about this. These are big projects that involve multiple labs and, you know, in some cases 50 co-authors and stuff like that. But we might be on the brink of finally, whenever I said in this talk, like, and this allows us to isolate it to, you know, a single branch or something. What I'm hoping is, I mean, I'm always thinking, yeah, why don't we do that? And then I'm thinking, oh, because in some cases, we don't have the tissues. We know this fungus was collected in Panama, and it's the sister to all the higher fungi. It's the lower sister to the higher fungi, but we only have like a tiny amount of it, and we have to go out and get more. And I'm always telling people, like, hey, when you're in Panama, get more of that. But you know, it isn't like you know where to get it. You have to. So, but I think we're getting close. And I do think, of course, there's going to be, well, okay, there's another answer too that I need to tell you. Even in that, when we did that fungal phylogeny, and we had ultra-conserved elements, right? We were able to determine the function. These are just chosen almost at random, these ultra-conserved elements. But we were able to determine the function of many of them. We extracted the protein coding regions of these things, and then we looked them up in these, you know, protein tables and stuff. And we found that many of the functions definitely kind of corresponded to, I can't tell you, I wish I could tell you what the functions were right now, but I can't. Things like degrading lignins and stuff like that. But previous studies had already identified many of these genes in other contexts in the fungi. So we knew we were onto something. So we do have clues like that. But I think those things, I think it'd be much, I think when we have whole genomes, I think we're going to find gene complexes that have to do with growing fungi. And in the fungi, we're going to find, like in the yeast fungi, we're going to find something in there that has to do with the yeast phase. It's going to be really great. More.
Like that, there is a mention of certain amino acids being in higher proportions.
Oh yeah, that's in. Oh, well, you're asking the wrong person. How do I Oh, here. I'm figuring out how to go back to that. Do you want me to go back to it? Yeah, okay, so, oh, wait, okay, so the question is, one of the slides mentioned amino acids. And like, how is that, in what way is that connected to fungus farming? That slide showed what I was calling the domestication traits of higher fungi. And it was the last thing, and I didn't mention it, but there's up leveling of certain amino acids in higher fungi. And I don't remember what the amino acids are, but I can tell you that all fungus farming ants, unlike all other ants, have lost the ability to make arginine. So all other ants can make it. Fungus farming ants can't. I'm pretty sure that wasn't one of the amino acids that was being up leveled, but presumably the fungi are supplying the ants with arginine. But I can't tell you specifically what the other, but I'm sure, I mean, I think it must be related to the symbiosis.
Thank you very much, Ted. Thank you all. We still have 40 minutes in the room, so if you wanna stay, ask more questions to Ted, hang out, talk, and we're gonna have our summer recess for the next few months, and we're gonna go back to the regular business meetings on the first Thursday of October.